Good morning, and thank you for everybody attending this congress. Now we have closing lecture by Professor Mariusz Ratacjzak, who is a very special friend of mine and also a very special friend of the Brazilian PURINES Club. So the presence of Professor Ratacjzak honors us very much. I would like to introduce briefly Professor Ratacjzak. He is the Henry and Stella Endowed Chairs of the Graham Brown Cancer Center at the University of Louisville. He is currently, among many other distinctions and awards, he is the editor-in-chief of Stem Cell Reviews and Reports. He has received the Presidential Distinguished Faculty Award for Outstanding Research and Scholarship of the University of Louisville. He has the Karl Landsteiner Life Achievement Award, the German Society for Transfusion Medicine and Hematology, and he is honorary professor of German University of China, and he has received the Doctor Honoris Causa of the Medical University of Silesia. Professor Ratacjzak is a very famous scientist. He is an international known specialist in the field of adult stem cell biology. He has discovered embryonic-like stem cells in adult bone marrow and other tissues, which have the potential to make a revolution in the field of regenerative medicine. Professor Mariusz has done important contributions to purinergic signaling in hematopoietic stem cell mobilization. They are also defining ATP as a danger-associated molecule, a damp molecule. He has worked on purinergic signaling in the inflammation and the innate immune response. Professor Mariusz has an H factor of 76 and more than 24,000 citations. Professor Mariusz, this is an honor to have you here, and I want just to remember the audience that you click below the session and you go into the Zoom mode, and this Zoom mode will allow you to make answers, questions through the question and answer boarding by chatting, or you raise your hand and we'll call you to make your question. And just remember that my co-chair is Professor Claudiana Lamil. Professor Mariusz, thank you very much, and please go ahead with your lecture.
I would like to thank very much my dear friend and respected collaborator, Professor Henning, for this nice introduction. And it is really very big honor for me to present this lecture for PURINES Club of Brazilian Society. I will be talking today about purinergic signaling in hematopoietic stem cells, but so truly not so much about effect of purines on proliferation, because it was a very nice work by Professor Francesco Di Virgilio and Roberto Lemoli, who were working on a role of purinergic signaling in proliferation of hematopoietic stem cells. Instead, I will be talking about a role of purinergic signaling as a regulator of hematopoietic stem cell migration, which is highly relevant for hematopoietic transplants. And hematopoietic stem cell transplantation is well-established life-saving medical procedure, and its success rate depends largely on number of transplanted stem cells and speed of their engraftment after infusion to the patient. Therefore, clinical outcomes will benefit from improving strategies to harvest stem cells from the donor and accelerating subsequent homing and engraftment of these stem cells in the bone marrow that they will establish a healthy hematopoiesis. And here are shown most important steps. And in every of these steps, what I will be showing you, purinergic signaling is playing a very important role. First of all, right now, very common what we perform in the clinic, it is with hematopoietic transplant using mobilized peripheral blood stem cells. On this first slide, you can see here a donor who received some drugs like granulocyte colony stimulating factor, GCSF, or CXCR4 inhibitor, AMD 3100, which move stem cells from bone marrow into peripheral blood, and stem cells are collected by apheresis. Next, a recipient of a transplant is undergoing very high dose of chemotherapy, which destroys pathological hematopoiesis. And then hematopoietic transplant is just intravenous infusion of collected from donor cells into the recipients, and these cells sense a gradient where they have go to bone marrow and engraft. And purinergic signaling is playing very important role in all these three steps, and we can modify these steps by modifying purinergic signaling. And in my talk today, I will focus on pannexin and P2X receptors because are specific for ATP, extracellular ATP, and also I will focus on ectonucleotidases and final product of adenosine, which is activating P1 receptor. And here at this slide, you can see our data. We have a phenotype human, but similar data we have for murine hematopoietic stem cells for expression of P1 and P2X receptors. And you can see hematopoietic stem cells, also like mononuclear cells, but hematopoietic stem cells in particular, very highly express P2X7 and P2X4 receptor. For P1 receptors is very high expression of A2A and A2B receptors. And what I will be showing you today is an effect of ATP adenosine yin-yang effect in stem cell trafficking, where ATP is activator of this trafficking, and CD39, CD73 product adenosine has opposite inhibitory effect. It is like yin-yang effect according to Chinese philosophy. And first, a few words about retention of stem cells in bone marrow and the role of membrane lipid rafts, because it will be important to understand next part of my lecture. Hematopoietic stem cells are anchored in bone marrow stem cell niches, and here is very important interaction of CXCR4 chemokine receptor expressed on hematopoietic stem cells with ligand stromal derived factor 1, which is expressed in stroma cells. And what is here very important, CXCR4 receptor for SDF1 is membrane lipid raft associated receptor. It is enriched in special domain in cell membrane, enriched for cholesterol and sphingolipid. And here you can see an example showing human CD34 positive hematopoietic stem cells. It is here confocal picture. We stain these cells for expression of CXCR4 receptor, shown here in red. And then also we stain for membrane lipid rafts. Here is shown in green, and here is yellow colocalization. And you can see CXCR4 is an evidence, is membrane lipid raft receptor. Why it is important? Because if CXCR4 receptor is included into membrane lipid rafts, is much better connected to downstream signaling proteins, and interaction of SDF1 with receptor is stronger. This means cells much better respond to stromal derived factor 1 when CXCR4 receptor is included in membrane lipid rafts. And there are some priming agents doing this. And as you will see, extracellular ATP is one of these agents, which promotes incorporation of CXCR4 into membrane lipid rafts. And what is significance, biological significance of this interaction? First of all, if cells see SDF1 gradient, which is very important attractant for hematopoietic stem cells, as is shown here in transwell membrane assay, chemotactic assay, and they see SDF1 together with priming agent, like for example, extracellular ATP, they migrate much better to this gradient. They are hungry for this gradient. And they, as is shown here to the right, much better migrate, respond to SDF1, much better home to the bone marrow. And now what is here important, bone marrow stressors release extracellular ATP as a major purinergic signaling mediator that triggers activation of one of pattern recognition receptors, intracellular pattern recognition receptors, which is NLRP3 inflammasome. And it is because of sterile inflammation. Sterile inflammation is a common event triggered by physical, chemical, or metabolic damage stimuli. It's not related to microbial infection. And most important, pharmacological mobilization, what I was showing, on one of the first slides, as well as conditioning for hematopoietic transplantation by radiochemotherapy to destroy old hematopoiesis and make place for new hematopoietic cells, is causing sterile inflammation in bone marrow. And this activates purinergic signaling, NLRP3 inflammasome, and complement cascade axis. And here is a very well-known scheme, extracellular ATP by P2X7, but also P2X4 receptor activates this intracellular complex, which is NLRP3 inflammasome. And this inflammasome cleaves pro-interleukin beta and interleukin 18, two mature interleukin 1 beta, interleukin 18, which are secreted out of cells. But at the same time, caspase also cleaves gasdermin to make a gasdermin pores in cell membrane, and that release several alarmins from the cells, including, for example, high mobility group box1 protein, also more extracellular ATP, and S109A. And it is activation of inflammasome. And here are our data where we have shown that inflammasomes, this innate immunity pattern recognition receptor, is highly expressed not only, as for many years people believed, in innate immunity cells, that means granulocytes, macrophages, but is also expressed in human and murine hematopoietic stem cells. And now, hematopoietic stem cell mobilization, this means egress of stem cells from bone marrow into peripheral blood, where they are harvested later by leukapheresis to prepare graft for a recipient, is regulated by purinergic signaling. And here is first, we looked what is a role of pannexin 1 channel. We know that it is a major channel which releases ATP to extracellular space and promotes ATP to potent purinergic signaling molecule. And in this experiment here, we have inhibited in mobilized mice, pannexin 1 by peptide, blocking peptide. It is specific pannexin 1 blocking peptide. And here the most important, here shown in this red area, that number of hematopoietic stem cells released into bone, to to peripheral blood from bone marrow after administration of granulocyte colony stimulating factor is decreased here in mice which were exposed to pannexin channels. This means pannexin is important in the release of ATP into extracellular space. Then ATP is interacting with P2X receptor, and based on our data, P2X4 and P2X7 are very highly expressed on hematopoietic stem cells and are playing here very important role as activators of NLRP3 inflammasome. And as you can see here, we have mobilized P2X4 deficient mice, and they are poor mobilizers here in these red circles. As you can see, number of hematopoietic stem cells released after mobilization with GCSF and MD is decreased in P2X4 deficient mice, in contrast to wild-type animals shown here in red squares. Now we look for P2X7 deficient mice. Again, these mice are poor mobilizers in response to granulocyte colony stimulating factor. And then signaling from both receptors merges on NLRP3 inflammasome. And here, NLRP3 inflammasome or wild-type animals were mobilized, as you can see, with GCSF or MD3100. And as you can see, in comparison to wild-type animals, mice which don't have NLRP3 inflammasome, they are poor mobilizers. Then we looked for downstream of NLRP3 inflammasome for caspase-1 knockout animals. And as you can see here, again, caspase-1 deficient mice are poor mobilizers after stimulation with GCSF or MD in comparison to wild-type mice. Then we ask, okay, if here activation of inflammasome releases alarmins or danger-associated molecular pattern molecules like high mobility group box1, S109A, and also additional extracellular ATP. And on the other hand, two very important pro-inflammatory cytokines, interleukin-1 beta and 18, which of them are very important to restore a proper mobilization in caspase-1 deficient mice. And here, as you can see in these experiments, again, wild-type mice mobilize, very nicely. The effect is in caspase-1 deficient mice. When we have substituted caspase-1 deficient mice with recombinant interleukin-1 beta 18, is not much effect, but it was effect when we added DAMs, danger-associated molecular pattern molecules. This means high mobility group box 1, extracellular ATP, and when we have added S109A. And the same experiment here in NLRP3 knockout animals, which don't activate caspase-1. Again, as you can see here, we were able to restore proper mobilization in presence of danger-associated molecular pattern molecules or alarmins. And what are doing DAMs? DAMs activate complement cascade. You can see here we have injected to mice a cocktail of DAMs, alarmins, and as you can see here, we were able to detect in these mice activation of complement cascade. It is detection of C5a component, cleavage fragments of complement cascade. And it is, we are coming back to some data before we started to work on purinergic signaling. Before I met Professor Hennig, who actually brought me to this field, we have shown that complement cascade deficient mice, C5 deficient mice, are poor mobilizers. They mobilize very poorly with GCSF, as you can see here, in comparison to wild-type animals, white here bars, and also in bone marrow and in histology, as you can see, capillaries in bone marrow are empty in C5 deficient mice in comparison to wild-type animals mobilized with GCSF. And what we propose a model is that stress stimuli induce sterile inflammation in bone marrow. And then ATP is released from some innate immunity cells, some hematopoietic cells, stroma, and activate NLRP3 inflammasome. And this cogwheel here, NLRP inflammasome, by releasing danger-associated molecular pattern, molecules activates complement cascade. And complement cascade downstream from purinergic signaling, NLRP3 inflammasome is crucial in release of stem cells out of bone marrow. And as you can see here, we have summarized here on this slide that, after administration of GCSF-MD, innate immunity cells, if we don't have immunity cells, are poor mobilizers because they don't release ATP. If we don't have Panexin-1, working Panexin-1 is also poor mobilization. Everywhere where is red asterisk is poor mobilization. If we have defect in P2X4, P2X7 receptor, poor mobilization. Defect in expression of NLRP3 inflammasome, caspase-1, and complement is leading to poor mobilization. Now, what happened in next step in hematopoietic transplant? Purinergic signaling is also very important, that hematopoietic stem cells harvested from donor infused to the recipients, to the patient, they sense properly hemotactic gradient and go to the bone marrow. And again, we were looking on Panexin channel, what happened when we don't have Panexin channel. And as you can see here, if we blocked by small peptide, Panexin channel on hematopoietic stem cells, here is control peptide, here is Panexin-1 channel blocking peptide, we saw decrease in early and late engraftment, showing here in comparison to wild type cells, which were unmanipulated, in terms of showing number of PKH2 labeled cells which home to the bone marrow 24 hours after transplants, to colonies, chronogenic progenitors after 24 hours, similar 11 days after transplant progenitors were impaired homing when these cells before transplant were exposed to Panexin. And what is here important, hematopoietic recovery here was also decreased. It is recovery over time of leukocytes and platelets, and it was decreased if Panexin channel was blocked on hematopoietic stem cells. And to explain this, what is very important, Panexin channel, as you can see, blocks lipid raft formation on hematopoietic stem cell. This means Panexin is blocked, ATP not released, and we don't have lipid rafts, which make cells hungry for homing SDF1 gradient. Yeah, what I am showing here is that extracellular ATP sensitizes hematopoietic stem cells by increasing incorporation of CXCR4 into membrane lipid rafts, that they will go better to bone marrow, sense better homing gradient. Now we look what happened when we don't have P2X receptors. And again, if we have P2X4 knockout hematopoietic cells, as you can see in comparison to wild type cells, is shown here black bars, P2X4 knockout hematopoietic cells home much better to bone marrow in assay 24 hours after infusion, and also in assays 11-12 days after infusion to the recipient. And also, if we block P2X7 receptor, we have very similar phenomenon, decrease in early engraftment, early homing, red here squares in comparison to unhealthy wild type hematopoietic cells, and also red here in red is shown delay recovery of platelets and recovery of white blood cells in transplanted animals. And here again, to make this story complete, the same situation, if we have NLRP3 inflammasome deficient hematopoietic stem cells, they have also defective homing and hematopoietic recovery after recipients which got such cells have decrease in delayed hematopoietic recovery after transplant. Now, last part, what happened in microenvironment? As you know, a hematopoietic transplant recipient before transplantation is receiving high-dose radiochemotherapy to destroy pathological hematopoiesis. And this also induces in microenvironment sterile inflammation and also purinergic signaling as part of the sterile inflammation plays here a very important role. Again, we expose a recipient mice before transplantation of wild-type cells to pannexin-1 blocking peptide. And as you can see, if we blocked in recipient mice pannexin-1 in hematopoietic environment, again, early and late engraftment was impaired. And also in recipient mice, it was delayed hematopoietic recovery of white blood cells and recovery of platelets. Now, what happened, we know, with P2X receptors. And here, what is very important and important evidence that after lethal irradiation or chemotherapy as a procedure to prepare for transplant, inflammasome is activated in bone marrow 1 hour, 6 hours after conditioning for transplants. This means we have a state of sterile inflammation and activation of NLRP3 inflammasome. And here, as you can see, mice which are defective in NLRP3 inflammasome, they engraft much worse here in assay of homing and early engraftment with wild-type bone marrow hematopoietic stem cells. And to explain this, like purinergic signaling, effector, NLRP3 inflammasome. Here you can see wild-type mice in the middle, NLRP3 knockout mice, and these mice were conditioned for transplant by lethal irradiation. We could do the same with chemotherapy, and as you can see here, in NLRP3 knockout animals is much worse response in upregulation of SDF1, this homing factor to bone marrow, and KIT ligand, which is important stem cell factor for expansion after transplantation. And again, NLRP3 knockout animals very poorly activate complement cascade. And we are coming also to our old data showing that mice which have defective complement cascade, they are C5 deficient, is one of the major complement cascade proteins. Distal part of complement cascade activation depends on C5, engraft much poor. They have delayed recovery of leukocytes and also of platelets. And finally, it has to be some inhibitory regulatory mechanism. And then we focus on P1 receptors and adenosine, especially on adenosine. And as you can see here, adenosine is very strong inhibitor. It is chemotaxis assays of hematopoietic stem cells to SDF1 gradient, but also bioactive lipids S1P, C1P, and also extracellular ATP gradient, which are very strong chemoattractants for hematopoietic stem cells. It is inhibited this migration in presence of adenosine. And here, if mice were mobilized, for example, as is shown in GCSF or MD, it was decrease of mobilization if we injected these mice with tolerable dose of adenosine. And to explain this, and maybe make here a little bit jump, what we have found that a very important inhibitor of cell migration is hemoxygenase 1. And ATP decreases in hematopoietic stem cells level of chemooxygenase 1 at messenger RNA, and here are shown in panel D protein level by Western, and also adenosine has opposite effect, upregulates hemoxygenase 1. And what we here would like to postulate, ATP released from mononuclear cells through P2X7, P2X4 receptor activate NLRP3 inflammasome, pattern recognition receptor of innate immunity, which stimulates cell migration. But at the same time, ectonucleotidases, by metabolizing ATP to adenosine, upregulate hemoxygenase 1, which inhibits cell migration. This means we have here the yin and young effect on stem cell migration between extracellular ATP and extracellular adenosine. How to improve this? Here is one of the possibilities. Mice before transplantation were exposed to CD39 inhibitor, and we improve homing of hematopoietic stem cells by lowering extracellular ATP in bone marrow. The same when we inhibited CD73 ectonucleotidase in bone marrow, we also improved homing and engraftment of hematopoietic stem cells. And it is a last slide here showing, how we envision navigation, migration of hematopoietic stem cells to bone marrow chemoattractants, mostly stromal delay factor, but also sphingosine phosphate and also ATP release from the bone marrow. Is activation here of NOX2, which upregulates ROS. Reactive oxygen species are very strong activators of inflammasome. Then, extracellular ATP is released from hematopoietic stem cells as alarmin, and in autocrine manner activates P2X7, P2X4 receptor to propagate this autocrine loop. On other hand, CD39, CD73 express on hematopoietic stem cells work on ATP, making, extracellular adenosine, which then on retroacting age activates A2A, A2B receptors, which are highly expressed on hematopoietic cells, upregulate hemoxygenase 1, which then by negative feedback inhibits inflammasome and stem cell migration. Conclusions of my lecture today is that extracellular nucleotides, most important extracellular ATP and adenosine, are important regulators of trafficking of normal stem cells. ATP released from the cells into extracellular space activates through P2X7 and P2X receptor. It's very important signaling mediator, NLRP3 inflammasome and caspase-1 in hematopoietic cells. And while extracellular ATP activates an LRP3 inflammasome and stimulates migration of stem cells, the zinc effect is metabolism. Extracellular adenosine has an opposite effect. And stimulation of ATP and LRP inflammasome axis will improve stem cell mobilization and homing after transplantation. By enhancing purinergic signaling, we can get more mobilized stem cells into peripheral blood. On vice versa, by inhibiting extracellular adenosine action or increasing hemoxygenase 1 signaling, we can also improve homing and mobilization. And I would like to thank you very much for attention. As this work mostly was done by Mariusz Adamiak, who is making right now habilitation, by Arjun Tapa, my postdoctoral fellow, Kamila, my collaborators here in my laboratory, but also it was a very important contribution of my respected and dear friend, Professor Henning Ulrich from University of São Paulo, and also other collaborators listed here, Dr. Ratacjzak, Dr. Kucia, Daryl Davis, and Amnel Abdel Latif. It was my pleasure to present this presentation. Thank you so much for your attention.
Professor Mariusz, thank you very much for this fascinating talk. Really great, and it shows you direct translational applications of purinergic signaling, right? ATP may promote or may be or is good for stem cell transplantation. So I would like to open for questions. So just to say, raise your hand or put the question into Q&A. So there is a question that I will read to you, Mariusz, yes, from Professor Hobson Coutinho Silva. Dear Mariusz, thanks for the excellent conference. A friend of mine who directs the Department of Bone Marrow Transplantation at the Federal University of Rio de Janeiro once told me that it's necessary to freeze collected bone marrow cells to have better results after transplantation. do you see convergence between what you showed us today and this empiric information? Can you comment with hypothesis?
Yes, it was actually excellent question, and most important observations coming from such observation in the clinic. How I envision to explain: freezing is a stress. Stress is releasing extracellular ATP, because by freezing, a lot of extracellular ATP is released into extracellular space. And this ATP, what is doing most likely, how I envision, is priming responsiveness of hematopoietic stem cells by incorporating CXCR4 receptor into membrane lipid rafts into the chemotactic gradient of SDF1. Yes, and we even are using a little bit different strategy that we are adding, and I didn't show this data, before transplantation on non-frozen bone marrow, on normal bone marrow, ATP, just to make these cells that they will vigorously and robust respond to SDF1 gradient, because ATP will promote incorporation of CXCR4 into membrane lipid rafts. And yes, freezing, thawing is doing the same job, releasing alarmins, releasing ATP, which is doing the same effect, incorporating in CXCR4 into membrane lipid rafts.
Mm-hmm. So thank you very much. I also have I would have many questions for this fascinating talk. No? So the first one was, if you say that CXCR4 is getting into this lipid raft, is the same thing with P2X7 and P2X4 receptor? Did you check this?
Is very good question also, because like I say like this, there are three major chemoattractants for hematopoietic stem cells. We have many different chemokines, whole laundry list, and different factors, but three more important, according to my many year research I was doing, chemoattractants is stromal delay factor, which is acting through CXCR4 receptor, extracellular ATP, of course, acting through P2X7, P2X4, and sphingosine 1-phosphate acting through sphingosine 1-phosphate type 1 receptor. All these receptors, CXCR4 is very well documented, is membrane lipid raft. Sphingosine 1-phosphate receptor is membrane lipid rafts. We have also some indications that P2X7 is also membrane lipid raft receptor. And I recall some papers, not on hematopoietic stem cells, but on cancer cells, where people reported incorporation of P2X7 into membrane lipid rafts. This means it seems very common mechanism. So, but we never did on hematopoietic cells, directly. We are planning to do these experiments.
Yes. So, and now, if you talk about membrane grafts, I'm also thinking into exosomes. So, would you think about that by CXCR4, for example, and for example, the P2X7, P2X4 receptor, that you may have a communication through microvesicles, now exosomes, which could line up or coordinate the activity of hematopoietic stem cell mobilization?
Yeah, it's a very good question. Actually, on Professor Francesco Di Virgilio, yes, he was showing ATP secreted, in microvesicles. It is in exosomes. Yeah, it is no well phenomenon. But in hematopoietic transplant, exosome role is very pleiotropic. First of all, we have reported a long time ago that if we take, for example, exosomes from the platelets, peripheral blood platelets, this exosome can stick and paint surface of hematopoietic cells with some adhesion molecules, which are very important in attachment of hematopoietic stem cells to endothelium, especially bone marrow endothelium, in capillaries. This means they make them sticky in bone marrow, that they can much better go to bone marrow, because if we transplant hematopoietic cells, only 10% to 20% reach bone marrow. Other stem cells are just lost in the periphery. This means painting of hematopoietic cells with microvesicles could be very important for homing to the bone marrow. of course, microvesicles are enriched in ATP and they can release also ATP. Also other factors. This means definitely exosomes modulate many situations in trafficking of hematopoietic stem cells. And it is very, there's also several papers showing that, stimulation of hematopoietic cells by exosome expands these cells, stimulate proliferation. We have published 20 years ago that exposure to exosomes improves homing to the bone marrow. Other groups follow up these footsteps showing similar phenomena. And of course, taking in consideration that exosomes have a lot of ATP, it makes everything sense.
So one last question. So you showed that the models of down regulation or knockout for P2X7, for Pannexin 1, and also for, I think it was for caspase 1. So if you, for example, and you use the opposite. So for example, now you would transfect for overexpression of P2X7, P2X4 receptors, hematopoietic stem cells, and then you make basically the transplantation. Yes, would you expect that you have a better result in mice?
Yeah, it is as usually very good questions. Always you ask, yes, yeah. Exactly, because if we expose what I was, first question was, about this freezing thawing, releasing ATP. For priming effect. And I mentioned, if we expose hematopoietic cells before transplant to extracellular ATP, they also engraft much better. If we stimulate, for example, NLRP3 inflammasome directly, which is a downstream target of purinergic signaling P2X7 and P2X4, and there are some, drugs when you can increase, level of NLRP3, you have much better homing. Definitely, here is a lot of also vice versa of strategies to improve homing. And last thing also that I was showing, if you modulate microenvironment in bone marrow, this way that you downregulate expression of extracellular adenosine, which is released during sterile inflammation as well by inhibiting ectonucleotidases, you can also improve homing and engraftment. This means there are different possibilities to modulate this process by directing purinergic signaling elements.
So last thing. So if you the sensitization that you get together in a lipid cleft and by ATP and adenosine would dissociate, yes? How do you think this is happening physically?
Yes, it is a good question. If we give more ATP, it is a risk we will have also adenosine. Yeah. No, it is here, at least empirical, but also, in experiments, we have shown that such procedure to expose to extracellular ATP improves. Of course, we have to take in consideration hematopoietic stem cells have on the surface CD73, CD39, CD73. They will make also adenosine. But, at least in our hands, this exposure to extracellular ATP is very powerful and it works.
Okay, so great. Is there any more question from the audience? Or
I would like to
yes, Claudia, please.
make a question. Professor Mariusz, do you consider a receptor assembled with P2X7 and the P2X4 units in this mechanism or not? Because some papers show it's possible to assemble purinergic receptors with P2X74 and P2X7 units.
Yes, it is, a very, very important question you ask, because, it is like this, that thanks to great work, seminal work of Professor Francesco De Virgilio, we know a lot about P2X7 signaling in different settings. P2X4 receptor, you would be surprised, is understudied. And for example, it was very recently, paper in journal Clinical Investigation, looking for a role of P2X7 signaling in leukemic niche. And they admitted, that, in contrast to P2X7, hematopoietic stem cells are also our data, highly express P2X4. And question, how important is P2X4? If P2X4, for example, heterodimerize P2X7, if they signal separately, it is still here, discussion, how it really works. But, at least in our hands, having access to P2X4 knockout animals through our collaboration with Dr. Davis, which are very unique mice, and also having specific inhibitor, we see a lot of effect of P2X4. And if it is true, what some investigators postulated, that P2X4 is much faster signaling purinergic receptors than P2X7, and that P2X4 is more sensitive to ATP than P2X7, because there were such studies published, then, we should start also to work on P2X4. Not only, alone P2X4, but also in collaboration, with P2X7 signaling. But anyway, this data I have shown you, expression of hematopoietic stem cells confirmed recently in this JCI paper, hematopoietic stem cells express very highly P2X4 and P2X7 from all P2X family receptors. This means definitely you ask a very important question, and more studies are needed, because we did a lot of beautiful studies on P2X7 to ask a question, what P2X4 is truly doing. And we are collaborating here with Professor Hennig, on this topic, to address how important is P2X4. We see some differences, we submitted paper recently in speed of signaling, which confirmed, data that P2X4 is much faster signaling receptors than P2X7. But it is still too preliminary to share because it was not still reviewed and it's still not published. But I encourage to look for P2X4, especially in hematopoietic cells, leukemia as well.
Okay, thank you.
So, Mariusz, thank you very much. And also your continuous
It was my pleasure. It was
contribution.
my pleasure and honor. And I still remember that I am also member of this society.
Yes, you are a member. Yes.
But I owe you probably fee because I didn't pay last year because of this COVID. You have to send me a bill.
Yes, we do this. Yes, we talk about this. Okay. Yes, but
Okay.
you are so very welcome, and we thank you very much for your contribution. Yes. And
Thank you. Thank you once more. Thank you.
So I will now, in a couple of minutes, we will go to a closing session. Yes. And I invite all of you to the closing session. Yes, where will also be where some will be communicated the awards of this congress to the poster presentations. And I would like to thank you, and I hope to see you in a couple of seconds. Thank you.